All right, so why don't we get started? First of all, it goes without saying that I hope everybody's doing well, and I'm sorry for the stress and everything that everybody's under. Ready? All right, here we go. Okay. Nice. You guys, if you understood what we talked about last time in terms of the strengthening of connections, this lecture will be pretty easy to understand, and it'll just be kind of an interesting extension. If you weren't 100% clear, I think hopefully this session will help you to become even clearer. To remind ourselves of where we were, you've got these two neurons, and we start off with synapse 1 is the strong connection because it's got obviously more AMPA receptors. There's none in synapse 2. Both neurons fire at the same time. Obviously, this neurotransmitter, this glutamate molecule, all it has is an NMDA receptor to bind to. These can bind both to NMDA and AMPA receptors. Since the AMPA receptor at this point is what's going to depolarize the neuron, as you recall, any depolarization that we get is thanks to synapse 1. That is to say, thanks to the firing of neuron 1. Right now, neuron 2, basically it's almost non-functional. I mean, in a certain sense, it's not functional in the sense that you can't activate neuron 3 based upon neuron 2 alone, but you can activate it based upon neuron 1. That's our scenario. As you recall, if all of these neurotransmitters from both synapses bind to all the available receptors, you get depolarization thanks to synapse 1, and we're going to represent that as the multicolored lines, which is then going to spread to the entire neuron. That's not the only thing you need. You need that strong depolarization, but you also need glutamate binding to the NMDA receptor at a particular synapse. Well, that you get thanks to neuron 2 firing. And that means that when we get plasticity, which we will in this case, you can have an increase in AMPA receptors at the synapse. That wouldn't happen if you got depolarization, but you didn't have any glutamate binding to the NMDA receptor. So you need those two ingredients. All right. And as a result, you now get an increase in AMPA receptors, at whatever synapse had depolarization, which was all of them, plus NMDA receptor binding, which was both of them. What happens to memory in the brain if an increase in AMPA receptors doesn't last long? So suppose the brain doesn't have normal long-term potentiation, and that is, suppose you've got neurons that maybe they produce no AMPA receptors, but they don't hang around for very long. What if those new ones only last for an hour instead of a week? Well, in rats, you can actually do manipulations, genetic manipulations, so that they don't have long-lasting LTP. Or you can have animals that are older that just through the aging process, the long-lasting LTP became really just short-term LTP. And what happens is the old animals are unable to produce long-lasting LTP in the hippocampus, and they also have poor memory compared to the younger rats. And the ones that have the biggest impairment in the LTP are the ones that have the big memory problems. So let's imagine a concept neuron. So imagine this concept neuron that when it's activated brings to mind your friend Susan. So you remember you've got a concept neuron for various people that you know and things that you know, places that you know. So this one imagined to be the concept neuron that must become activated in order for your friend Susan to come to mind. It's receiving input from a neuron that carries information about Susan's face, and it also receives input from a neuron that carries information about Susan's name. In other words, you hear somebody say, "Oh, do you know Susan?" And just that word, that name Susan, activates this neuron. Maybe it's connected to auditory nerves, which is going to, it could potentially at least activate the concept neuron, which would bring Susan to mind. And in this case, when you see Susan's face, that would activate this neuron. Maybe it's connected to the visual facial recognition areas. And when this neuron's active, that could also bring Susan to mind. That is to say, it can activate the concept neuron. So now, according to the diagram to the left, do you think that Susan's face brings to mind Susan? Now, I can tell you that these bulges here represent an increase in the number of AMPA receptors. So this synapse has got a lot of AMPA receptors. Give it a guess. Okay, great. It would be because Susan's face activates this neuron that we're going to call the Susan's face neuron, releases glutamate. It's got plenty of nice AMPA receptors waiting for the glutamate, and you're going to get a nice depolarization, crosses threshold, makes this concept neuron fire, and all of a sudden you're thinking of your friend Susan. Great. So in our other way of diagramming it, Susan's face releases glutamate. You've got lots of nice AMPA receptors here, depolarizing the neuron, causing it to fire, and you think of Susan. She comes to mind. So does Susan's name bring to mind Susan in this scenario? Why not? Why does Susan's name not bring to mind the idea of Susan here? So it's not strong enough, this synapse, and so Susan's name can't activate this neuron. That's correct, as Afan put it. There's no AMPA receptors, as Alyssa said. Well, that's exactly why we say it's not strong, because there's no AMPA receptors. So according to the diagram, hearing Susan's name does not bring to mind the idea of Susan. Why not? Well, we just said there's not enough AMPA receptors. So the concept neuron, this guy, responds to seeing Susan's face because when the Susan's face neuron releases glutamate into the synapse, it binds to lots of what? What type of receptors? And you know, and by the way, you know, there's going to be an AMPA receptor, an NMDA receptor or two in this synapse as well. But the reason we're focusing on AMPA receptors is because we're talking about the face neuron being able to activate the concept neuron. So the concept neuron responds to seeing Susan's face because when the Susan's face neuron releases glutamate, it binds to a lot of AMPA receptors on synapse 1. Susan's name doesn't activate the concept neuron, so it doesn't make you think of your friend Susan, because Susan's synapse 2 doesn't have enough what? Okay, not enough AMPA receptors. And so there's going to be no way to let sodium in and to depolarize the neuron. So in order for synapse 2 to get more, again, what is synapse 2 going to get more of? What do we want more of? Exactly. You want to strengthen the synapse, it's AMPA, right? I think actually, to be honest, AMPA to me, actually, if you're talking about something that's strengthening a synapse, AMPA sounds like a good name. Seems stronger than NMDA. Maybe it's just me. If you didn't see Susan's face, and the only thing that happened is somebody says Susan, and you're not thinking of Susan, weak synapse. So they say it again. Susan's, you know, somebody says, do you know Susan? I don't know. It's not bringing her to mind. Wait, just think. Susan, 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 Susan. That sounds like a pretty obnoxious person, right? So no matter how many times you say the name Susan, if you don't have any AMPA receptors, you're not going to strengthen the synapse. In order for synapse 2 to get stronger, You need more AMPA receptors to appear at synapse 2. And there's no way for that to happen just from hearing her name, because you'd be releasing glutamate. There'd be no AMPA receptors for it to bind to. You know, you might have an NMDA receptor here. You probably do. But the neuron's not going to be depolarized if there's no AMPA receptors. You need the neuron to be depolarized at the same time that the glutamate is going to bind to the NMDA receptor. So you could have glutamate binding, but you would not have the depolarization needed to remove the magnesium.
I was going to mention that in order to form an association between both concept neurons, you would have to see Susan while hearing her name. Is that correct?
Yeah, that's correct. But the way we're going to put it in this scenario, you're exactly right, is that not a matter of more, we're just imagining one concept neuron. Like you're imagining you've got this concept neuron that represents Susan. It represents her name, it represents her face, it represents the type of dresses she likes, anything you know about her that would bring her to mind, we'd say that's because of activating the concept neuron. So in a sense, what we're going to do is the change that we're going to make is, instead of her face being able to bring her to mind, it could be her face that brings her to mind, or now we're going to see that it can be, and we'll see how, that her name brings her to mind as well, brings her to your mind as well. Yeah, Isamar.
Oh, yeah, I got it. So in other words, you have one concept neuron, and it's basically receiving input from the neuron that's Susan's face and a neuron that's Susan's name, correct?
Exactly. Yeah.
Thank you.
I mean, you know, if you say visited her home a lot, and you know, were saying, well, what if I just see her home? What I think of Susan? Well, same thing. Imagine you've got a neuron activated by seeing her home, and ask whether that would be strong enough to activate the concept neuron. That's essentially what we're saying. You know, can all these things that might bring Susan to mind, can each of them bring her to mind? Right now, the only thing that's bringing her to mind in our scenario is her face. Marcia, yes.
Hey, it's Marcia, by the way.
Marcia, I'm sorry. Yeah.
Okay, quick question. Why do we need an AMPA receptor to depolarize when the NMDA has a glutamate receptor?
It cut off at the end, but I think you were saying, why do you need the AMPA receptor when you've got an NMDA receptor? Is that right? You can chat it in if you want. Yeah, that's correct. Okay. Yeah. The reason is because with an NMDA, if you imagine an NMDA receptor here, and Susan's name releases glutamate, that glutamate, say, binds to this receptor. So what happens when glutamate binds? Well, the same thing that happens at other receptors, say an AMPA receptor, is it opens a channel. That's great. So now you've got the channel open at the NMDA receptor, and yet you still don't have any sodium or calcium flowing in, so the neuron's not getting depolarized. Why isn't it flowing in? It's because in addition to this gate, which is now nicely opened, you also have a magnesium ion blocking it. You're going to need magnesium to move out of the way in order to get sodium to come in, or calcium, okay? If you wanted to depolarize it just based upon the NMDA receptor, you'd still need to get magnesium out of the way. Well, you can't, because it's not depolarized. And no matter how much you say her name, since you don't have AMPA receptors, there's no way to depolarize it using the AMPA receptor. And the AMPA receptor is great because you can take a very hyperpolarized neuron, and as long as glutamate binds, the gate opens and you can get at least some depolarization. But it's hard to know how you get this thing off the ground, and the answer is going to be you can't without the help of this already strong synapse. Go ahead, Afan.
So is there always a magnesium attached with NMDA receptor and not AMPA?
Yes.
Okay.
Correct. There's always a magnesium that's bound, but in order for magnesium to be bound and blocking this channel, the neuron's actually got to be, relatively polarized, like -70. The magnesium doesn't have much affinity for this channel as soon as the neuron becomes depolarized.
Right.
So if you've got AMPA receptors depolarizing the neuron, now you can get magnesium to move away, and now, so long as glutamate binds, to open up the channel itself, the gate, once you've got depolarization, magnesium moves away, and then glutamate binding can allow depolarization, through the positive ions. Exactly. And AMPA receptors never have magnesium blocking them. It's just the NMDA receptor. And it's not like a defect of the NMDA receptor, right? It's basically a way for the NMDA receptor not to be too easily activated. Because, don't forget, when you activate the NMDA receptor, calcium comes in, activates some enzymes that are going to then enter into the nucleus of the neuron, cause new proteins to be made, create more AMPA receptors. Well, that's the whole, important business, strengthening a synapse. You don't want that to happen just every time glutamate binds to the receptor site. You need, to be strict. You say we need two conditions. Glutamate's got to bind to the receptor at the same time the neuron's depolarized, and then we'll strengthen the synapse. All right. So in order to get more of these, okay, again, so let's just go back. In order for synapse 2 to get stronger, more AMPA receptors need to appear on the dendrite. Hold on one second, Jade. Of synapse 2. In order to get more of these AMPA receptors, you're going to need calcium to flow through the what receptor at synapse 2? And then I'll come back to Jade. Exactly. All right. The NMDA receptor channel does allow calcium and sodium both to come in. But here's the thing. The important thing about the NMDA receptor really is that calcium can come in. That's what makes it special. That's what's going to give us our plasticity. You can have as much sodium as you want coming in. That's not going to cause the nucleus of the neuron to produce more receptors. It's the calcium that's the magical ingredient, really, that sets into motion this chain of events. So just to say, it is sodium and calcium that can come in, but calcium is really the special thing with the NMDA receptor that's going to produce our plasticity. And then, go ahead, Yancy. You said, is it always magnesium that will block the channel, or could it be another cation? It's always going to be magnesium. Okay, so let me just back up. Without calcium flowing into the NMDA receptor at synapse 2, new proteins can't be created in order to produce new AMPA receptors at the synapse. But when you hear Susan's name, here's where it gets interesting. At the same time that you see Susan's face, the glutamate from the face neuron binds to AMPA receptors at synapse. What synapse were we talking about? And this strongly what? Depolarizes the entire concept neuron. So in terms of the question is, well, how do you depolarize this neuron, so that the NMDA receptor's magnesium is gone? How do you do that if there's no AMPA receptors to depolarize it, and the NMDA receptor is offline while the neuron's at rest? And the answer is you do it with a little help from your friends. All these AMPA receptors allow nice strong depolarization so long as you see Susan's face. And then what's going to happen is that depolarization is going to spread from the area of the face input throughout the entire neuron. So imagine again that multicolored illustration spreading all along the cell body, down through the axon, up along all of the dendrites. You say all of the dendrites, does that mean it's going to activate NMDA receptors in every dendrite? Not necessarily, because you need two conditions to activate the NMDA receptor. You need the depolarization, which we're getting thanks to our face synapse, but you also need glutamate binding to that NMDA receptor. Well, that's only going to happen if you're hearing Susan's name at the same time, so that the glutamate can bind to that little lonely NMDA receptor here. So let's look at it like this. If you hear her name and you see her face at the same time, seeing her face. Let's say, so in other words, that's why you've got glutamate in both of these synapses. Glutamate binds to all of the available receptors. And what we want to know is, can we fulfill the conditions to activate this NMDA receptor and get more AMPA receptors? That's the key question. And we say, well, in order for that to occur, you're going to need glutamate to bind, because that's going to open up the channel. And that needs to happen at the same time that the neuron's depolarized, because that's going to cause magnesium to be released. So let's see what happens. Both of them are active. Thanks to the synapse 1, you get your depolarization through these AMPA receptors. The depolarization is spread throughout the entire neuron, including to our synapse 2. And if all you saw was her face, that wouldn't be enough. But if you see her face and hear her name at the same time, you've got depolarization at the same time that glutamate's binding to the NMDA receptors. Now you activate that NMDA receptor. Now calcium can flow through and send the signal. We're going to need more AMPA receptors here, guys. That's basically the signal, and you get them. So, to review this, when you see Susan's face, as you're hearing her name, the name Susan causes release of glutamate to the NMDA receptor at synapse what? At the same time when the concept neuron is already, well, depolarized. Exactly, Christopher. You got it. If you see her face as you're hearing her name, just to review this, the glutamate from the face neuron depolarizes the neuron, and that depolarization, this is the key thing, the depolarization spreads all the way to synapse, it spreads all the way to synapse 2. If you see her face as you're hearing her name, okay, because you also hear her name, glutamate's released, because you hear her name, that's this neuron, remember, glutamate is also released into synapse what? So it's being released into synapse 2 at the same time that the neuron's depolarized. So at the NMDA receptor of synapse 2, you have, number 1, glutamate binding. That's because you heard her name. At the same time that the magnesium ion's been liberated, thanks to the depolarization that was caused by synapse 1, by seeing her face. This means that what can pass through the NMDA receptor? Yeah, it could be calcium and it can be sodium, but the star of the show here in terms of plasticity is which? Exactly, calcium. You got it. That's our magical ingredient. And this calcium inflow through the NMDA receptor at synapse 2, now that you've got the perfect situation, yep, you've got the perfect situation for learning, this calcium inflow triggers gene expression and the synthesis of the, fill in this blank here. You got it. That's a $500 prize. That question was worth $500 for Chic. Synthesis of the one of the things, unfortunately, since everybody is microphone is off, and if I say something and anybody else laughs, I don't get that positive reinforcement, and so I just have to laugh to myself. So like that's what it's like here for me. That's all right. It was funny. Okay, yeah, that actually helps. The fun. Thank you. And synapse. Okay, yeah. That's interesting. If I make a joke, you can give me the thumbs up. Oh, what about, oh yeah, Michelle, yeah. If I make a joke, it's funny, it's not necessary. But, if you give me something like Michelle's emoji, it would be fine. That would be good. Yeah, something like that would be appreciated. Let me see what else we got here. How about, what about, something like this? Yeah, would it kill you to give me something like that? There you go. Yeah. Okay, all right. Let's get back to the plan here. So the calcium inflow triggers gene expression, synthesis of proteins needed for more AMPA receptors to grow at synapse. Where do we need more AMPA receptors? Synapse 2. All right. Now, so now finally we've got our new AMPA receptor here. I mean, as you can, imagine, you get a new one here too, because, all the conditions were met for more AMPA receptors here too, because you got, depolarization of the neuron and glutamate binding to the NMDA receptor here as well. But, I mentioned last time, there's going to be some upper limit on how many AMPA receptors can grow into the synapse. Now, do you know Susan? You say the name. I can picture her right now. Okay, so this is how you now are able to think of Susan by seeing her face or hearing her name, because you had enough experiences where you heard her name while you were seeing her face. And guys, if you want to, when you review this, to make sure like you really, really, really get it, is what you should do is you should be able to imagine that if you just kept seeing her face, why it is that wouldn't be enough to strengthen the name synapse. I mean, intuitively you'd say it shouldn't be, but in terms of these things we're talking about, why that shouldn't do it. And also make sure you're clear on why somebody can, if you're not looking at her face, you can hear somebody shouting her name to you 100,000 times and you will never strengthen the synapse. Make sure as you're looking through it that that's clear to you why that shouldn't be enough to make her name bring her to mind.